Human-pig hybrids, chickens with dinosaur legs, and plant cells injected into animals. These are terrifying hybrid creatures scientists created in labs. GloFish are probably one of the most famous examples of genetically modified pets. These little fish, which are actually zebrafish, were engineered in 1999 to glow in the dark. The trick? Scientists inserted a jellyfish gene into the zebrafish, causing them to produce a fluorescent protein that makes them glow under ultraviolet light. The idea behind the glowing fish was actually pretty practical at first. The researchers wanted to use them as a way to detect water pollution. If the water was contaminated, the fish would change color. But as often happens with things that are cool and unusual, people started getting more interested in these things as potential pets. And by 2003, GloFish were available for sale in the United States, and they quickly became a novelty item. But their release raised some ethical questions. Some people thought it was just fun, but others were uncomfortable with the fact that these were pets that were cooked up in a lab. Plus, there were worries about what might happen if these fish were released into the wild, where they could potentially disrupt ecosystems. GloFish are legal to sell in many places, but they are banned or restricted in others. At the Mayo Clinic in Minnesota, scientists injected human stem cells into pig fetuses. What they ended up creating was something that could be described as the first Pigman, or a pig with human traits. The goal of the experiment was to study how human and pig cells would interact when fused together, because why not, I guess. What they discovered was pretty wild. Some cells kept their separation, so in the pig's body, there were areas with just human cells and others with just pig cells. But other times, the cells actually fused together and formed a brand new type of DNA that no one had ever seen before. So on the outside, it was a regular pig, but on the inside, it was something much more complicated. An animal with half human, half pig cells throughout its body. The most interesting part was the BLOOD, which was a hybrid of human and pig, and this was a completely new type of BLOOD that had never existed before. Scientists were able to confirm that this was the first time such an experiment had ever been done. Now this one's pretty mind-blowing. For a long time, scientists have been piecing together how dinosaurs evolved into modern birds. And in 2016, a team of researchers in Chile set out to watch this evolution in real time by experimenting with chicken embryos. Their goal was to look at how the leg bones of birds have changed over time and how they connect to their dinosaur ancestors. Dinosaurs, like the ones we see in fossils, had two long bones in their lower legs, the fibula and tibia, which ran parallel to one another. Over millions of years, those bones evolved. When it came to modern birds, the fibula became much shorter and developed a splinter-like end, while the tibia became the dominant bone in the leg. Now the researchers wanted to see if they could make chicken embryos grow that older dinosaur-like leg. So they took a gene that plays a role in this leg development, the IHH gene, and suppressed it in the embryos. And the chicken embryos developed leg bones that looked a lot more like dinosaur legs, with both bones being of similar length and reaching down to the ankle. These dino chickens didn't survive long enough to hatch, but this was the first time scientists could directly observe how bird legs evolved from dinosaur legs in living creatures. Before this experiment, it was just all speculation from fossils. Back in 2018, researchers produced sheep-human embryos. They wanted to see if they could grow organs inside these engineered animals. The end goal is to have body parts that could one day be used for organ donations. Scientists introduced human stem cells into sheep embryos, and while the embryos were mostly sheep, a small fraction, less than 1%, contained human cells. Stem cell biologist Hiro Nakauchi from Stanford University said, "The contribution of human cells so far is very small. It's nothing like a pig with a human face or human brain." And yeah, it was not that, fortunately. Yeah, not a huge percentage here, but the fact that they were able to create sheep embryos with even a small fraction of human DNA is pretty wild. The sheep embryos were destroyed after 28 days, so no sheep-human hybrids were actually born, but the experiment showed that creating organs inside animals with human DNA could be a possible solution to the growing need for organ donors. Oh, Florida, the state has enough wild stuff to worry about than, super snakes, but hybrid pythons are a real problem in Florida. These snakes are massive. Some could grow up to 23 feet long and weigh nearly 200 pounds. Originally, a lot of these snakes were exotic pets, but over time, a lot of them were released into the wild, and by the late 80s, the Burmese pythons had grown into a pretty sizable population in Florida and became a massive problem for the ecosystem. But even worse, researchers studying the pythons in 2018 found something pretty unsettling. Out of 426 snakes they sampled, 13 were hybrids. These hybrids were a mix of Burmese pythons and another species, the Indian python, which had also been released by pet owners. The two species hadn't just recently mixed, they'd been interbreeding for a long time before researchers discovered them. The Indian python is smaller but quicker than the Burmese python, and this combination makes them tougher and more adaptable. In 2009, researchers in Russia and Belarus created genetically modified goats that could produce human breast milk. It wasn't exactly pure human milk, but it was close. It contained 60% of the proteins that make up human milk. Not long after, a team in China took things even further and created a whole herd of 300 cows that could also produce human milk. The idea was to be able to harvest human milk on a large scale, mass produce it, and then sell it in stores. I mean, they figured people would not only want to buy human milk, but human milk that came out of a genetically modified farm animal. The Chinese team originally planned to have their milk in stores by 2014, but the idea didn't exactly catch on as they'd hoped, and the plan got delayed. Back in 1996, a group of scientists at Harvard, led by Joseph and Charles Vacanti, created what became known as the Vacanti Mouse. This mouse had a human-shaped ear growing right on its back. Now, this wasn't some weird mutation, it was a planned experiment. They took cow cartilage cells, placed them into a mold shaped like an ear, and then implanted it under the mouse's skin. The ear-shaped mold helped guide the cartilage into the desired shape, and as time passed, the cartilage naturally grew into that shape. The goal was to see if they could create artificial ears on mice and then graft them onto humans. The mouse used was what's called a nude mouse, an immunocompromised species that doesn't reject foreign tissue, so it was perfect for this kind of work. In an interview with Newsweek, Joseph Vacanti joked the mouse had the ear removed and then lived out a happy, normal life, which wasn't true. Lab animals are typically euthanized after experiments, and the original paper mentioned the mice after sacrifice, a term used to describe when animals are killed post-experiment. Ilya Ivanov was a Soviet biologist in the early 20th century who became infamous for his attempt to create a human-ape hybrid. Back in 1910, Ivanov gave a talk about the possibility of creating a human-ape hybrid by using artificial insemination. Then, in the 1920s, he took his theories to French Guiana, where he carried out a series of experiments. He tried inseminating female chimpanzees with human genetic material, but this was unsuccessful. After that, he went back to the Soviet Union and started trying to organize similar experiments, only this time he'd flipped them around, injecting ape genetic material into human volunteers. These plans were thankfully interrupted when the last of his orangutans died. Ivanov's work wasn't just controversial, it was downright disturbing to many, and it led to his eventual arrest and exile. Just this year, scientists at the University of Tokyo managed to combine chloroplasts, which are green energy-producing cells found in plants, with animal cells. The idea here was to give animal cells the ability to create energy from sunlight, a process called photosynthesis, just like plants do. For years, scientists had tried to mix chloroplasts with animal cells, but the animal cells would just digest the chloroplasts within a few hours. That was until the Tokyo team used a specific type of algae called this thing here. But this specific algae could stay active in animal cells. They inserted these chloroplasts into Chinese hamster cells, and to their surprise, the chloroplasts didn't just survive, they kept performing photosynthesis for up to 2 days. The hamster cells even grew faster than usual. Looked like the chloroplasts were giving them some extra energy. Researchers still aren't sure exactly how the chloroplasts and the hamster's mitochondria interact, but they're working on it. And these Planimal Cells could possibly help us find new ways to create tissues or even new forms of energy. In 2019, reports came out about a team of researchers from the Salk Institute in the United States who'd successfully created the first human-monkey chimera. Led by Professor Juan Carlos Psua Belmont, the team had already produced embryos with small amounts of human cells in pigs and sheep, but this time they went a step further, working with monkeys. The idea was similar to their previous work to create chimeras that might one day be used to grow human organs that could be used for life-saving transplants. These human-monkey chimeras were reportedly only allowed to develop for a few weeks though. The process was carefully monitored and stopped before any of the embryos developed a nervous system or organs, which could raise more serious ethical concerns. These chimeras were also created in China to avoid legal restrictions in other countries. The difference in evolutionary distance between humans and monkeys is much wider than between humans and chimpanzees, but it still opens up the possibility of growing organs for transplants. The chances are slim though that human-monkey hybrids could be brought to full term because of the vast amounts of genetic differences between the species. All that said though, I've been your host James, and I'll catch you, yes you specifically, in the next video.